be in the way.
Right on. Cool. So what is a mushroom? So scientifically, mushrooms don't really exist, really. It's dependent on whatever phyla you're looking at. And that's only really 2 phyla of fungi that we know about. There's like 9, 11, 4, I don't know. It doesn't really, it depends on who you're asking. But today we're going to be going over 2 because those are the ones that do like mushrooms, how do you call them? So mushrooms are filamentous structures from fungi. They are the reproductive structure, and they
Like they say, there are exceptions to every rule.
that is very important. That's going to come up like 10 times in this presentation because it's absolutely true. Especially the more that you study biology, the more that you see that like the things that you've been taught are only like 90% true. Yes, and cool. Yeah, so this is the taxonomical tree of fungi. Like I said, there's debate on how many phylum there actually are because a lot of them are kind of similar, but we're only going to be focusing. So we're only going to be focusing on these 2. So it's Basidiomycota and Ascomycota. Cool. But it's still important to know about the different phylum, just from an evolutionary standpoint. And as you can see, the most primitive phylum is Chytridiomycota. And side note, when it comes to scientific classifications and terms and everything, Latin's a dead language, so it doesn't really matter if you like pronounce things right or whatever, but they're also built with prefix signs. So if it's like intimidating, just break it down. Chytridiomycota. But
say it with confidence.
exactly, yeah, exactly. That's the most important thing. And those grow only in aquatic systems, so they're found everywhere from lakes to ponds to soil to the bottom of ocean vents. And we know that they're primitive because they have flagellated gametes, similar to animals. And that's not something that you see in the later phyla of fungi. And those are most popular because they kill amphibians. They've caused the extinction of, I think, like 100 different amphibians in the past like decade or so. And it's a really, really big issue that we need to deal with. And
there's research happening at Texas State on that fungus that's in the I think it's in Panama. Like it's the same fungus that I think Merlin Sheldrake, who wrote that book, Tangled Life. Like he was looking at that too, but people have told me we should have that professor come talk because he's
yeah, that'd be awesome.
Yeah. Sure. Like I don't know anything about it. I just know like when I first read about it, it's in a book about the Anthropocene or the coming Anthropocene. And they talked about this frog hotel that they've created in Panama where they have been able to keep it sterile and keep this fungus out. I'm like, that's
yeah, I heard about that. I think it's the same like you have to dip your shoes in alcohol before you can walk into the hotel. Just make sure that you're not tracking any of the I'm gonna go hot. Okay, that's fair. And also another thing with Chytridiomycota is that they don't have hyphae or mycelium as we see them. They have rhizoids, which pretty much act as anchors. They don't grow how we typically see mycelium. And then after that, you have Zygomycota, and that's where you start seeing hyphae coming into different fungi. And I'll get into what hyphae is in a bit, but these are most typically just molds that you see on bread or various foods. Those are most likely going to be Zygomycota. Not always, but mostly. And then you have Glomeromycota. Kind of slipped up on it, it's consonants. And those are mycorrhizals. That's whenever you start seeing the hyphae actually interacting with plants and the environment. And additionally, fungi produce spores, not seeds, obviously. Here's a picture of, I'm not exactly sure which one it is, but some puffball that I found a few years ago. And you can see it's producing a gust of spores as I squeeze it. So as I said, fungi are composed of little tubes of chitin. So plants have cellulose that they use to produce their structure and rigidity and protect their cells. And fungi use chitin. It's very similar, but it's I think the extra glucose in there or something, and it acts. It just gives it much more rigidity and strength than normal. And you can actually see this whenever you're cooking, because if you ever cook mushrooms, they just kind of like shrink because they lose their water, but the actual structure of the mushroom is very, very stable. There's actually been very recent studies that it can withstand temperatures up to 400 degrees Celsius, which is pretty crazy. And also chitin is what composes insect exoskeletons. And that's why fungi are able to so easily eat that, is because that's already what their cells are made out of.
I like your illustrations. Can you do those?
Yeah,
There's
a few in them.
Yeah, I like the color. Yeah.
I'll also get more into, because you can see there's like red and pink and purple and stuff. I'll get more into like what that actually means as it goes on. Those are nuclei. So remember that, but I'll get into the specifics later. So here's a diagram of the different cells. So as you can see, all three of these are eukaryotes. So they're all going to have nuclei. They're going to have mitochondria, Golgi apparatus, but there are slight differences. So in this center one, that's us, animals, we have a plasma membrane, and that's why we're squishy. Oh. Yeah, that's why we're able to be like very flexible and goopy. And plant cell walls, they have a cell wall that's why they're not very bendy. They can kind of sway in the wind a little bit, but maybe it'll break and snap. And that's just how they evolved to protect themselves, as opposed to being squishy, I guess. And fungi have the best of both worlds because they have a rigid cell wall, but it's also extremely flexible because of the extra glucose that is in the cell wall. And here you can see fungus cell that's budding, which we'll get into that later. And here's just a diagram or some pictures of some cordyceps taking over some bug and some cordyceps from The Last of Us. Dog seen that show?
Yeah.
It's very good, very good. I haven't seen the last episode.
That just looks like AI art.
Yeah. I stole that off Google, so I'm not exactly sure what bug that is, but I just thought that the way that it resembles a skull was very, very cool and very topical because fungi eats things. So there's exception to every rule, but fungi generally are composed of hyphae, which are very, very small structures that you can't actually see with your naked eye. And this is just how this organism has evolved to maximize surface area, because we're all doing that in our own different way, maximizing surface area while minimizing the energy needed to take in that energy. But yeah, fungi produce these webs. And this is called the surface area to volume ratio in biology. If you want to look more into that, because like I said, every single, even same organisms in different species, it's fascinating. So with fungi, you start off with a spore, and the spore is just the most basic unit when it comes to fungi. They're single-celled, and they're reproductive, and they are haploid. Here you can see some Rhizula. I'm not sure which species it is, but the spore print actually blended in very, very well with the postcard. So I had to use some glue to make it visible. And it just goes to show that spores come in all different colors and everything. So adapt. So haploid. What that means is that these cells only contain one set of chromosomes. And to make it easier to understand human, we have two sets of chromosomes throughout our entire lives. It's called diploid. And the only time that we have haploid cells is our reproductive cells. And that is different with fungi, where they can live out a vast majority of their lives as haploid because they only have one set of chromosomes. And that is usually denoted by the lowercase n. So the spores will branch out into filamentous structures called hyphae, which are haploid. And this is the part that is actually entering the plant's cell walls whenever mycorrhizae is happening. Here you can see there's actually two different types of mycorrhizae, where it's ectomycorrhizae and endo, and that just means that it's penetrating the cell of plants. But they both act in the same symbiotic way that mycorrhizae do. So once the hyphae grows enough, it becomes mycelium, and this is the part that you can actually see with your eye. And mycelium is very interesting because it can either be haploid or dikaryotic. And dikaryotic is in like vocab word because that is unique to fungi. There are not any other organisms that we know of that are able to produce or to undergo dikaryotomy, or sometimes called heterokaryotony. And I'll get into what that means in a little bit. But as you can see, the mycelium on this little sculpture dude has begun growing to the point where you can really see it covering the entire thing.
Is that like an art project or was it?
No, it was I was growing some blue oysters and I just got bored with it one day and I just kind of like molded it out of curiosity to see what it would do. And it started growing and I was like, oh, it's funny. No actual mushrooms grew from it. I'm assuming it's because it wasn't that much. It was only like this big. I'm not sure, but no, it just goes to show the potentials of because
And the substrate is just like some like grain or something.
yeah, brown rice flour was good. Yeah. And side note, again, to show how strong chitin is, people build or use chitin, use mycelium as building material. And that's kind of what this became. Since it didn't really fruit or anything, it just kind of became a dry little sculpture. Yeah. All right, so mating types. So typically, whenever you're talking about mating types in fungi or in horticulture or breeding fungi, you use plus, minus, sometimes A, B, lowercase, capital, 1, 2, but it's generally plus or minus. And that's just to make things easier because we understand sex to be a binary, usually. And the dependency of the mating type is actually on two chromosomes or two alleles rather, on the fungi's chromosome. And they're called alpha or beta, depending on which one. And usually there's two variations. There's alpha 1, alpha 2, beta 1, beta 2. But again, there are exceptions to every rule. So we're very, very dependent on species. Some of them have 3. Schizophylum commune has 300 variations for alpha and 90 variations for B. So it has 200 compatible mating types. That's usually what you see online saying like, oh, well, this mushroom has 200 different sexes or 200 different genders. Gender is very wrong because that's yeah, for sure. That would not even be close to being right. Sex is more accurate, but even that isn't super accurate because
it just likes a lot of
yeah.
I'm gonna say it's skewed.
It's very compatible is the best way to put it, a little flirty,
Spores.
Yeah, and that's usually the most common one because it actually has some benefits or a lot of benefits because the more compatible it is with different members of its own species, the more likelihood it has at reproducing. So for example, humans, and generally we have 2 sexes, and this means that we have a 50% compatibility with our population, but Schizophylum commune, I think it has like a 96% compatibility just because there's so much variation. And the only thing that matters is, I'm gonna cool. Yeah, here we go. Yeah, so here's just a visual demonstration of it. So the alleles, they're on this, it's called a locus, where the alleles are next to each other. And yeah, as you can see, there's alpha 1 and alpha 2, and beta 1 and beta 2. And it doesn't matter that the alpha 1s on this top one are matching because the betas are different. And that's all that they're looking for. It's just compatibility and genetic diversity. And yeah, you see that because these are compatible, they're able to undergo crossing over during reproduction and independent assortment, which is where genetic variation comes from. So as I was saying, usually humans denote sex as a binary, but that's wrong. It's very important to think like a scientist, and that means doing away with all biases or preconceived notions that you have, because science is about learning the truth, but that is very open to the data that you're looking at, and preconceived notions that you can have just going to interfere with data, just to be blunt. And humans try to categorize nature into different boxes, but that's the reason why exception to every rule exists, is because nature doesn't fit in boxes. Nature just is trying to, even that, nature's not trying to do anything, but nature generally is trying to just reproduce and grow. If you want to get real technical, we're just experiencing entropy, but science, as we know it, is just explaining or interpreting the natural world with our sensory organs. But as we can see from optical illusions, our sensory organs are very limited. There's no actual way to objectively define what something is. And we can see that through the history of science, where we think we know something, a new theory comes out, and it changes. And it's very important because of that to see science as something that is fluid and open to change as new data comes out. There's never going to be something that is hard set. I would say maybe evolution is an exception to that rule, because every single bit of evolution or every single evidence that we go towards that just reinforces it. But yeah, like I said, that's just something to keep in mind when it comes to understanding the world. So recap up to this point, because it's already been a lot of info. So mushrooms, as we have them, they are composed of hyphae, and the mushroom is actually the fruiting body of the fungus. And mycelium and the fungi are produced of hyphae, and the hyphae is produced of chitinous cell walls. You're kind of going in a pyramid style here. So here is the generalized life cycle of fungi. No, I said generalized because there are many fungi that do not follow this, but generally you will start with spores that are haploid, which will germinate, form mycelium, and they will undergo something called plasmogamy, which is whenever it's essentially the first step in mating, whenever two fungi of compatible mating types meet and fuse their cytoplasm. And they undergo the heterokaryotic stage, which I said is unique to fungi. And I'll get a little to more of that in a little bit. But then after they go from there, they will fruit and produce a fungi or a mushroom. And from there, the mushroom will undergo meiosis to split its DNA in half and have a new set of DNA for the next generation. So plasmogamy, as I said, that is the fusion of the cytoplasm. And whenever it comes to plasmogamy, generally that only happens during sexual reproduction, whenever the nuclei are ready to fuse. But with fungi, they are able to undergo heterokaryon, which is their nuclei fusing, or their nuclei entering the same cell, but not fusing. And that is not something that we know of normally. Normally, cells meet, they exchange genetic information, and then they have their offspring. But they're actually able to do this for years, stay in this stage where they have two complete distinct sets of DNA. Once the proper fruiting conditions come about, they produce the mushroom, and the mushroom is actually where the genetic combination happens, a very specific part of the mushroom. And like I said, humans are diploid. So we have two sets from the moment that the sperm and the egg fuse to the moment that we're dying. We are diploid. We have two sets of chromosomes, other than whenever we are making gametes. And I don't know if I said it, gametes are just the reproductive cells of whatever organism you're talking about. And sexual reproduction has the benefit of genetic variation. And genetic variation is very important when it comes to protecting yourself from the environment. Just the more variation there is, the more likely they're able to survive as a species. So plasma, this is the dormancy stage. So as I said, they are two sets of chromosomes per cell. And here's a diagram to better illustrate what's going on. So these are two compatible mycelium that have fused, and the new mycelia that they're making have two complete sets of chromosomes. And that's
So pause a second.
Yeah.
Somebody
Sure.
on the Zoom asks, what does nucleus just Netflix and
so they're just hanging out, they're not actually undergoing any kind of reproduction yet. They're just getting to know each other, I guess is a good way of putting it, where they because normally the nuclei of two organisms only ever meet whenever they are ready to reproduce. But this this is a unique stage of just hanging out.
friend zone?
in a way, temporarily. Are there
Any other questions on Zoom?
Yeah, so far. While
We're good,
while
that was
talking.
it.
Yeah, so the secondary mycelium here has two complete sets of DNA. And this is denoted with N plus N. And as I said, fungi, as we know, are the only organisms that can do this. I think there may be some protists that also do it, like slime mold type organisms, but don't quote me on that. I think it's just fungi, as we know. But like I said, there's an exception to every rule, so would not be surprised at all if there were other organisms that can do this. And generally, the nuclei are separated cell to cell by what's called septa. And you can see here that it's just a way of blocking off, it's like a filter that separates cells, but not all fungi have septa. Basidiomycetes, which is actually the most common mushroom that you usually see, don't have septa. They just have plasma that goes throughout the mycelia, readily spreading nutrients. And even with this, there's a exception because new research is coming out that there are some fungi that are able to be polyploidal, where they have 3 or even tetraploidal, 4 complete distinct sets of nuclei per cell, which crazy. How are they doing?
They've been around the block for a while.
Yeah, I guess so. They know how to get around. So Basidiocarps, this is the fungi that you normally see whenever you're out and about. As I said, mushrooms don't really exist. So the basidiocarp is the scientific term for what the mushroom is. And they can come in very huge variety. Here's a reishi that's shelping, a yellow oyster that's shelping with gills, some honey mushroom that is gilled with the typical umbrella shape that's still growing in clusters, and this jelly fungi that is just doing its own thing. No distinct shape or anything. And even spores, that's just kind of a general term. Whenever you're talking about spores, it depends on what phyla you're talking about. So Basidiomycetes always produce basidiospores. And you'll see, the more that I talk about this, they're really just using the same suffix with a different prefix, depending on what phyla it is. So basidiospores come from basidium on the basidiocarp. The basidiocarp is the mushroom. The basidiospores are spores, obviously, and I'll get into the basidium in this. So, basidium. So this is where the actual fusion of the nuclei happens. Sometimes it's called the zygote. I have a diagram in a second, but the mushroom actually, once proper conditions are met, they will form this basidium, and from there join their nuclei and then separate it into 4 unique combinations of the parent's nuclei.
That's under the microscope.
Yes, this is an electron microscope of what the basidium looks like. And here's a diagram to show the nuclei in the mushroom still have not fused, and that's how it is throughout the entirety of the mushroom until it gets to this point where they decide. I'm not entirely sure what. I'm sure there's a mechanism that triggers the basidium to grow, but I'm not entirely sure. But once that happens, the nuclei fuse, and then they split in half to 2 genetically identical daughter cells of the fused nuclei. So these are the exact same. And then from here is when they split in half and they mix up their DNA. So you end up with 4 genetically distinct spores at the very end.
So you have like multiple sort of mycelium put together, then like the resulting thing might have multiple different pairs of chromosomes.
For sure. Yeah, it's definitely going to. It's only going to be compatible with fungi of the same species. Again, that's kind of an asterisk because there are some people that are able to reproduce, kind of mate, I guess, different oyster mushrooms that aren't they're in the same genus, not the same species. But the actual reproduction, the mycelium, if you do it in an agar dish, for example, and you cut out the piece where the 2 myceliums have mixed, that mycelium that's growing will be genetically different from the 2, but it won't actually mix its DNA until a mushroom forms from that mycelium.
So say you have
Acids.
three different mycelia.
Sure.
Then like could you end up with a new mycelium with cells that were not homogeneous?
You're getting ahead of it. That's actually a phenomenon that is very new research that I'm not going to get into super, but I will answer that question later on in the slide. So good foreshadowing.
Talking about kind of like, you know, these other things that may determine like the fusion. These are the right terms, but there's all then the environmental factors, right? So there's something else happening, and I guess is all of the sort of communication. I've heard that like the mycelium is sort of the immune system, and it's detecting all the environment sort of things happening in the environment, so it knows like when the time is right to reproduce. So
Yeah, so
like sending those as signals.
Yeah,
things happening.
the environmental factors come into play when it comes to fruiting mainly. And when it comes to the actual plasmogamy or the fusing of the 2 different mycelium, as long as it's like they're in a substrate that has water, that's really the only requirement that, and nutrients, that's really the only requirement that matters. But environmental factors for sure come into play when it comes to fruiting, which comes into play of the mixing of the chromosomes, which I also get into the fruiting in a little
That's
bit.
way down the line. We're just talking about sport.
Yeah. And here's a diagram to show mushrooms. Some basidiomycetes can have the umbrella shape but still be polypores, and most of them have gills. And you can just see here the basidium in green dropping the spores.
Shooting through the pores.
Exactly. And it's actually very precise, the distance that the spores are shut out from the basidium. And that is because of something called Euler's drop.
Whoa.
So this, every single spore has something called an apiculus, and it's pretty much where the spore attaches to the sterigma, which is so in the basidium, there's 4 little udders, and those are called sterigma. And whenever the spore has matured, so as the spore is maturing, the center of mass is in the center of the spore. But once the spore is actually matured, the apiculus will start producing sugars, which attract water. And this little drop will start forming. This little drop will start forming at the base of the spore. And as that's happening, also the relative humidity of the environment causes the spore to be covered in just a small layer of water. And as that's happening, the Euler's drop is also growing. And once it gets to a certain size, there's a contact point. And because of cohesion, which you can see whenever 2 puddles of water go into contact, they kind of, yes, exactly. The water rushes to the tip of the spore. And it's actually that rushing that knocks the spore off of the sterigma. And the only reason that that's able to happen is because, again, of the chitin. Because the chitin is so rigid and strong, the sterigma is able to maintain its structure as opposed to just like kind of flopping around. Because if it did that, then it would lose all momentum, the spore would just stay. So very, very cool. So this is the generalized life cycle of Basidiomycetes, which may make a little more sense now. So we start off with spores, and the spores will germinate into mycelium. And once the mycelium meets its compatible mating type, they'll undergo plasmogamy, which is where the cytoplasm of the two mycelium will actually fuse and enter their dikaryotic or heterokaryotic stage, where they have two distinct sets of nuclei in the cell. And then once the fruiting conditions are appropriate, they will fruit. And then the zygote in the basidia will fuse its nuclei, and they undergo one round of meiosis to split the two daughter cells. And then they undergo a second round of meiosis to make four genetically distinct offspring. And then from those, you get spores. That's how it works. You start all over.
So the reason they're like matching hyphae.
Sure.
Like, can you explain that a little more? Because there has to be matching spores, and then the spores turn into hyphae. At that point, they match too.
Yeah, so all that it matters when it comes to the hyphae are that the information is different genetically, because some mushrooms are able to reproduce asexually. But as long as the hyphae are of different types. So there's two alleles, and it really depends on the species of mushroom. But as long as, even if half of the genetic information is the same, for example, two spores from the same parent, they would still be able to reproduce because they still do have varying genetic information. It's just, maybe more susceptible to environmental factors because of inbreeding and, the issues that may bring.
Yeah. So.
If not, I'll clarify. Cool. So fruiting, like you were asking about, this is extremely dependent on the species of mushroom. The only thing that we can really say that combines most of them is something called a hyphae knot, and that happens through evaporation. The hyphae will actually come up with the water that is being evaporated, and they'll form these tight knots. And once conditions are right, from there, they will form the primodia, which is actually very cool because from the start of the mushroom growing, they already have all of the cells that are in the mature mushroom, but they're just dehydrated. So they will, once the pin actually starts, they will soak up water from the mycelium and actually kind of grow, kind of similar to a human's reproductive structure. Also, actually, it's actually very, very similar to how they grow. And it's totally dependent on the species of fungi. There's different requirements, very, very subtle requirements also, like some fungi that can only grow once there's been a landslide and the mountain is at a specific slope. Very, very specific stuff. But some new research in the past 10 years has shown that there's some fungi that actually have light sensory proteins in a similar way that plants do. And it's very interesting because the light sensory proteins that these fungi they're looking at produce only picked up on green wavelengths, and plants pick up on everything except for green. So very, very weird, maybe just a coincidence, maybe not, who knows. But there's some research also from Japan that shows that logs in nature that have been struck by lightning are more prone to fruiting, and they reproduced that in a lab where they literally just shocked mycelium with fungi, and they found that the fungi they did that with was much more prone to fruiting. And I think that came out literally like a few months ago. So it just goes to show that when it comes to fungi, a lot of research is being done and needs to be done still. So it's good to have as many in mind as possible.
Those light sensors, those proteins are the same one that absorb UV that then convert it into like nutrients and vitamins. Right?
In a way, yes, because that is what the light sensory proteins do in plants. Whenever the wavelength hits, because there's different proteins that pick up the specific wavelengths, and once the electron from the wavelength hits that protein, it like jiggles it, and that causes energy pretty much to make it super, not thorough explanation. And they don't know if that's the same thing with fungi. It couldn't necessarily. It could not necessarily that it's getting nutrients that way, It could be that it is part of like the metabolism, where once they pick up on these lights, they know to grow in a different direction or something. We really have no idea. We just know that they exist, and even the fact that they exist is super, super new. So more research definitely needs to be done. We have no idea what the reason of those are. There are hypotheses, but those mushrooms, I think, also were able to grow in complete darkness. So I don't know if it would be nutrients. But...
I'll try.
Hello?
Somebody have a question on Zoom? Somebody just had their sound on by accident. Someone
And
did ask.
yeah.
Okay. So plasma.
Yes.
Homogeny occurs only with compatible mating types, question mark. So this will slide back.
Yes. So that is how they are able to fuse. They send out pheromones, and from the pheromones, if they are compatible, then they're able to fuse. And plasmogamy is it's how every organism reproduces. The cells need to, because if you break it down, it's just like cytoplasm, and then ogamy, which means like joining of, and normally, like I said, that only happens in other organisms when they are reproducing, like the final stage of, okay, we are going to have a organism or a life for the next generation. Let's fuse ourselves. But with fungi, they're able to do that and literally just hang out for decades. Yes, but that is dependent on the mating types. And yeah, when it comes to fruiting, there's so many exceptions to this rule. It doesn't even bear saying that this is how you fruit a fungus. Pretty much the only thing we can say for certain 90% of the time is that evaporation is required. So once the mushroom or Basidiocarp in this case has produced these spores, it undergoes something called apoptosis, which is just regulated cell death. And different fungi, again, have different ways of doing this. One of the most popular examples, one of the most studied examples is these Caprinus species or genus. The species are very, very similar. So I just kind of say Caprinus, but these are more commonly known as inky caps. And once these fungi reach maturity, instead of dropping their spores, they'll actually melt, or deliquesce is the term. And they use the water from the cells of the fungi to help the spores germinate for the next generation. And for that reason, they're one of the most successful mushrooms. I think they're on every continent except for Antarctica.
Anytime it rains, so like
Exactly.
the first ones to pop up. Mulch.
Exactly. Very, very common lawn mushrooms. Yeah, mulch, beds, everything. And they're so cool to look at, especially whenever if they're mature while it's still raining, because you can, it was a little dry whenever I took this picture and they kind of curled up, but
So cute when you look at eyelashes.
yeah. And they have little faces. But even with this, we don't really understand the mechanisms behind it. Like I said, we know that they have enzymes that will trigger once the fungi is mature enough that cause it to die, but we don't know the exact mechanisms behind it or anything. So still more research needs to be done with that. Research with that would be beneficial because understanding what specific things kill fungal cells would aid in fighting fungal infections. And we can use those same enzymes for as crop dusts, for example. Take that with an asterisk. I'm not saying that we should do that necessarily. Just more research definitely needs to be done, but just as an example of what the possibilities with more research. Cool. So that was Basidiomycota that I was talking about, and now we're going to move on to Ascomycota, which is a good way to kind of remember these. It's just the weird looking fungi because they don't really fit any specific shape. Here are some cordyceps. They're growing out of some deer truffle that they have this stalk with this head at top. But they're another very common Ascomycota is morels, and they look totally different. Another common one is the cup fungi, the diagram that I drew earlier. That's another Ascomycota, totally distinct. Exactly. Yes, exactly. And the way to differentiate the mushrooms isn't by how they look, but how they reproduce. So the Basidiomycota have the Basidium, but the Ascomycota have an ASCII, is what it's called. And a way that you can remember that is by the phrase "blow it out your ASCII," because they also don't have a Duller's draw, but they actually use osmotic pressure. So if you've ever seen videos of people blowing into cup fungi and then like a second later it puffs out, that is because the breath that you're doing is rupturing the cells.
I'm fascinated by fungi. I just did a little
Yeah,
video.
yeah, he posted it earlier today. Yeah, I literally saw it today and I was like, oh, that's perfect.
Your ass is.
Yeah.
I wish it was that little catchphrase.
Yeah, it's a really good at remembering what these do. And unlike Basidiomycota, Ascomycota are capable of sexual or asexual reproduction. Well, some of them. Here's a diagram to show. So you start off with ascospores instead of basidiospores, and it's going to be very similar to the previous life cycle, just with a few differences. These spores will germinate and find their compatible mating type, undergo plasmogamy, and form what's called an ascocart instead of a basidiocart. And same thing, where their reproductive structure is where they actually fuse their nuclei, and from there they're able to pass on genetic information to the next set. But what's different about this phylum is that they are capable of asexual reproduction.
Yeah, which I think in Japan they found our state mushroom, the Texas star. They found the asexual variety more recently, which is exciting.
Yeah, no, it's definitely one of the cool things with fungi that there's benefits to both, and I'll get into that in the later slides. But what they're capable of is just mind blowing, honestly. And here's a diagram. Or here rather is a bisection of, I want to say it's an electron microscope of the ASCII. So there's a very specific number of spores. Generally, they're going to produce 8 spores instead of the 4 spores of the Basidiomycetes. And as you can see, they're maturing from this red to this darker color. And once they have reached maturity, the top ruptures from the pressure and they all shoot out.
All of them at once.
All of them at once.
Wow, so pretty. And so that like a change, like I read like a change in relative humidity or like some sort of change in the environment, like maybe more moisture or something like that will then that's when things expand or something happens where then they fire off.
Yeah, it's probably like the wind is like a huge factor for these, like actually, because one common factor with a lot of these Ascomycetes is that they have the kind of cup structures where it like sinks into the mushroom, and that is to aid exactly that's the exact purpose. Yeah, yeah, that's why Ascomycetes are also sometimes called cup fungi for that reason. Sac fungi for the that's actually what ascus means in Greek, is sac. Or cup fungi, just because of the structure they have. Also, side note that I forgot, Basidium is Greek for club, because I guess they thought that the Basidium looks like a club. But yes, the Ascomycota are also capable of asexual reproduction, which is pretty straightforward. The mycelium or the spores will germinate into mycelium, and they'll produce these structures called conidia. And the conidia pretty much just replicate the DNA to spores. So all of these spores will be genetically identical clones to their parent mycelia. No actual combination or anything's happening here. And here's another electron microscope image of what these conidia actually look like. Pretty funky little trees. Some nice hair. And asexual reproduction has its pros and cons. In most organisms, a lot of energy is spent towards finding a reproductive partner, but with asexual reproduction, you could just, have fun. But of course, this comes with the con of no genetic diversity. So if something were to happen where a sickness were to come or a drought were to come and the genes weren't meant to fit with that, then it would just wreak havoc on the entire population. And there's actually some studies to show that because there are some lizards, for example, that their entire species reproduces asexually. There's no sexual reproduction that happens. And there are some hypotheses, rather not some theories, where this asexual reproduction will result in population explosions, but inevitable extinction. And we've never seen this happen, so that's why we don't call it a theory or anything, but we have seen trends with various species where they are asexual, something similar happens. So the pros and cons for sure, but it's very cool that these fungi are able to do both, depending on what is favorable.
Is there any studies too that have with maybe a kind of like this type of reproduction happening when there's like a lack of diversity maybe to begin with?
That's usually the reason why asexual reproduction happens, is that they are in an environment that is not favorable for a multitude of reasons. I don't think there's studies to show. Yeah, I don't think it's like necessarily like after X amount of time happens, they will reproduce. I think it may just be whichever comes first, either compatible mating type or optimal fruiting conditions. But with the asexual reproduction, they don't even produce the fruiting body, the ascocart. It's just little structures that come out of mycelium that kind of look similar to mold. So definitely pretty cool. Additionally, Ascomycota are able to, so yeasts, as we know them, are just a term for a single-celled fungi. Typically, whenever we talk about yeast, we're talking about like brewer's yeast or baking yeast, but yeast just means single-celled fungi. And yeasts are, instead of reproducing through filamentous structures and finding compatible mating types, they are only asexual, asterisk, and they reproduce by budding, which is similar to binary fission in bacteria. And all that they do is that they will get a chunk of their nuclei, and they will form this little cleavage here that separates the parent from the bud. And from there, the bud takes in nutrients and grows and repeats the cycle. There are some yeasts that are filamentous, which, exception to every rule. And for those, they usually don't, they're not as like motile, like these yeasts that are moving around in water and bumping into each other. Those will usually just drop a rhizoid or an anchor, and from there only produce the reproductive structure. So there's yeasts that are in the phylum Ascomycota, and they will have the conidia. But there's also yeasts that are in the Basidiomycota phylum, and those will just produce the basidium. No fungi or no actual fruiting structure or anything, which is, this is technically a fruiting structure, so the mushroom in a way, but yeasts are very cool. And also they are able of doing something called horizontal gene transfer, which this entire lecture has been to show how one organism is able to put its genetic information into another organism, but horizontal gene transfer completely cuts that away. And essentially, it's usually only seen with lower organisms like bacteria or some protists, but they're able to essentially inject their genetic information directly into the nuclei of other organisms. And here you can see an updated tree of life as opposed to the typical evolutionary tree that you normally see. This one has strands that are leading from lower class organisms to plants and some to animals and fungi, because they would not be where they were without horizontal gene transfer.
So that can be done like in labs using things like CRISPR and things like
Yes,
that, right?
exactly. It's very similar to that, but it's nature doing it by itself.
Yeah.
And that's actually the reason why we think that psilocybin is a thing, because there are various mushrooms or basidiocarps that will produce psilocybin that are not related at all. And one leading theory is that they are producing the optimal conditions for a bacteria, and that bacteria is producing the psilocybin and then using horizontal gene transfer to put that into the mushroom. Yeah,
Wow.
yeah, very cool. And yeah, this horizontal gene transfer, because whenever it comes to reproduction, it's only the very tip of the strand reproducing with another species organism in that same strand tip, but this is completely different genetic information. You know, this isn't like a zebra donkey. This is like a, you know, yeah. It's that, you know, times 1,000.
Yeah. And a
Um,
lot of the genetic information is just in the spore, right?
the spore has all of the genetic information that the rest of the mushroom will make. So as it's growing, each cell will have an exact copy of the original spore's genetic information. And that's up until it meets its compatible mating type, where they have two sets of nuclei. But it's the as far as the hyphae and mycelium are in their haploid stage, they will be exact genetic clones of the spore. I think that actually is a good transition into. Yeah. So to answer your question, and what you were just saying. So there's actually something, I didn't put it in this slide because I feel it was just the information overload at that point, but there is something called Buller's or Bueller's phenomena. And when that happens, we still don't, we've known that that's been a thing since the 60s or the 50s, but we still don't understand why it happens. And the fungi are actually able to combine their DNA and produce genetic offspring without making a mushroom or anything. So right in the cytoplasm, whenever the plasmogamy happens, they fuse, and then nothing else from there happens. But also at times, there's another thing called non-dikaryotic reproduction, where there is a mycelium that already has two sets of genetic information, and then there's a mycelium that has only one set of genetic information, and they fuse together and they swap their nuclei. So if it's 1, 2, 3, or 1, 2, and then 3, they will mix and then be 1, 3, and then 2. And we don't really know how that happens. So that's also new research, actually. That's only been observed. That specifically, the non-dikaryotic reproduction has only been observed the past few years. But yeah, Bueller's phenomenon we've known about for decades, but still don't really know what triggers, because, you know, what's the advantage of skipping the whole mushroom step and just doing it straight there. So pretty interesting stuff. And to recap, so we learned what fungi are. We learned that they have chitin as opposed to cell walls, and that they are filamentous structures that produce spores. We know that mushrooms now aren't really real, as we call them. They're either basidiocarps or ascocarps. We know that they undergo plasmogamy in their dormant stage, and they do that once they find a compatible mating type. We know what dikaryotic and heterokaryotic means whenever they have two sets of nuclei in the same cell, which is totally unique to fungi. We know that the genetic information fuses either in the basidium or the ascus. And we know that the basidium drops its spores because of Bueller's drop, which uses water as opposed to osmotic pressure. And we know that fruiting conditions are highly variable depending on the species, and we know what spores are. And that's the
Yeah, anybody else on Zoom, feel free to
questions or anything.
ask questions. But yeah, I feel like I've had reproduction of fungi explained to me a couple times, and this is by far kind of like the best
Oh, thank you.
explanation.
Thank you. I try to be thorough.
And I'm curious too, because I see your awesome illustrations.
It's
And I'm curious, like, what made you interested in, like, do you think that helped, like, drawing it out, like?
absolutely. When it comes to anything in specifically with biology, you don't, in my experience or in my history, you don't really understand what's going on until you can see it, because a lot of these things are so small, your brain can't really perceive like what is going on. But once you actually have, you know, thanks to electron microscopes, we know what's going on. And even those can be kind of limited because they're really just like an image as opposed to like a diagram. But I think the infographic type of information, you know, is very, very accessible to people, and it really helps visualize what's going on.
Yeah.
For sure.
colors,
All
shapes,
colors,
and
yeah.
sizes, yeah.
Use a little color theory in there.
Then look on the YouTube too to see if there's any questions there.
See if I-
Does anybody here have any questions?
Yeah.
There's a lot of yo.
So, so you have like mushroom, or sorry, basically, right?
Yeah.
And so you have another mycelium, they interact and they just like carry.
Yeah.
They have, you share their proteins. Somewhere else on the same mycelium, say there's another interaction with the mycelium, so that gets a different pair of proteins. How do they like reconcile this before they start putting on this? Like they always have consistent chromosomes across the whole set of mushrooms that are produced from this initial mycelium.
Yeah, very, very, very good question. That's one of the cool things about fungi is they are able to grow totally independently as long as they have a segment of their DNA. Like, for example, whenever you get a block of mushroom and you put that in your garden and it totally spreads, as long as there is a couple cells of DNA, they're able to reproduce. And because of that, the cells are separated by septa, like I said, and they're able to kind of think independently of each other. So even though the same organism will be mating with two different mating types at the same time, this one will produce a mixture that is totally independent of this one. Because even though like one of them is between the three, the partner or whatever will be different. And it's kind of just like, for example, I said that the septa is what separates the cells, but even in the Basidiomycetes where they don't have septa and it's just kind of like a fluid that's going between them, it still stays in the general area of the nutritional fluid that's going by. It doesn't go from like A to B to C.
And so cells that don't have like copies of this extra chromosome, do they just remain like with one set of chromosomes?
Yeah.
And then the ones with two chromosomes will like produce food body out of that cell.
Yep. And then as long as the organism is alive, it'll keep doing that. So the mushroom will grow and die, but the actual mycelium that has the genetic information, it will keep growing, it'll keep producing mushrooms, it'll keep mating with different types. And yeah.
It's just like experience. It doesn't seem like one organism.
No, and that's the
It's
really cool thing is that the way that mushrooms reproduce is totally unique when it comes to organisms. Like it would be the equivalent of like there's not really even equivalent when it comes to humans, you know. It's like imagine if like a sperm and an egg cell grew up their entire lives holding hands, and then they had a human, and then from that human they had another set of. It's great.
So you're doing agar work,
Yeah.
and you've got the mycelium that it's a matter where you're going to try to select a chunk of agar.
Yes and no, because there will be some mycelium that just aren't compatible, but I think there have been some independent studies that show that if you just keep growing the culture and keep trying, they will eventually be able to reproduce because of genetic mutations. But that is not necessarily guaranteed. If they're not compatible, and you can see whenever you're growing an agar, if there's like a little line between the two, or if they're kind of fusing together. But yeah, just better to take out the section where like, you actually see the two. Combine.
Usually you're waiting, there's quite a bit of mycelium too by the time you. I guess make a cut a section out, maybe.
Yeah, and that section that you cut out would be dikaryotic. So it would have two complete sets of genetic information, just waiting for the right conditions to prove.
Looks like there's a few more questions up there.
I don't think any plants produce psilocybin. I haven't heard of that I think of. But no, I think what you're thinking of is the bruising that comes whenever you will handle mushrooms that typically contain psilocybin. And they kind of think that that may be not necessarily like a defense mechanism, but something that is just like the cells rupturing. But we actually don't know the reason behind like why the psilocybin is produced. So it could be the defense mechanism. It could be something that helps the mushroom or the fungi grow. could be, that the bacteria is, we really have no idea. Definitely not for reproduction, though. I don't think. No. So maybe for defense, we don't really know, but there's a good chance that the mushroom itself is not producing it. It's bacteria. So you can think of this mathematically. So for humans that only have two compatible types, we have a 50% chance of reproducing. And say a mushroom that has 4 compatible types, like most mushrooms have 4 sexes, quote unquote, those have a 75% chance of reproducing with other species in their other organisms in their species. And like I said, the schizophyllum commune that has hundreds of different variations, that one has a near 100% chance at reproducing with another organism that it finds in nature. And well, it doesn't reproduce with humans, but another issue with schizophyllum is that it's very prone to germinating in your lungs. And yeah, so it causes.
There's so many spores already in your lungs, right?
Yes, but not all of them are capable of penetrating our cell walls.
Okay.
Yeah, so they may grow a little bit, but they'll just kind of die because there's nothing that they can do there. Yeah, exactly. But the schizophyllum commune is able to get around that, and it's not like guaranteed that if you handle them, that'll happen, because I think it's also the most farmed mushroom in the world, I want to say. And they're fine mostly. I think it matters on your immune system also, if you have a compromised immune system. And actually, there's been studies that show that people with compromised immune systems, the same thing happens with oyster mushrooms, where the spore is. Yeah, exactly. Yeah, when that happens.
You know, with farms, it's why a lot of the people wear respirators. And so there's gonna be lung issues, asthma, it gets aggravated.
For sure, nature does not care about anything. It's just trying to grow, and if it finds something and it's like, hey, seems suitable for life, it will start growing.
I guess there was one study, right, where they found the fungus, like, was it growing in the feet?
Oh,
Was
was it the little girl?
I think it's a male, but I can do a search. But yeah, it was like one of those clickbait things like that. Yeah. It's before last month's too. Like,
yeah, I saw one that it was a little girl that she had, I think it was schizophyllum commune, that was actually fruiting in her sinuses. It got to the point where it was growing. Yeah, really, really crazy.
Wow.
Because normally it just goes through the hypo stage, and then either the immune system kills it or it kills us. But yeah, it's like fruiting in her sinuses. Pretty wild to think of. Yes, so mostly yes, but there are still some strands of hyphae in the fungi that are going to be diploid or haploid of their parents. So here you can see how not all of these strands of mycelium that are going into the fruiting body are paired up yet. Some of them are just blue, some of them are just pink. And then here they're kind of like mixed up. Yeah, I would say the majority of the hyphae that you see are going to be combined of the two, but not all of them will be. So yes, mostly. Not 100%, but mostly yes. If you cut out a piece of fruiting mushroom and put it on agar, it'll be N plus N.
Try to repeat the questions.
Sure.
I can stand closer. Because I was gonna ask too, like, just out of curiosity too, like what brought you to like have this particular interest in fungi? Because I know a lot of people like they kind of belong on to like a certain area when they get introduced. And do you have like other areas that you're super fascinated by as well?
Yeah, for sure. So the actual reproduction came from one of the science classes that I took at community college. It was essentially the entire class was seeing how different organisms will reproduce. And I thought it was very funny because, we went over all the different mammals and like insects, and like, we went over flowering plants versus ferns and everything. And then we went over fungi, and it's like, where are the details? Where is like why aren't you splitting up into different categories like you are with plants or with animals? And I saw that as kind of a common trend where people will broad lump all mushrooms into just fungi, when in reality the way that they grow is super, super variable. And I guess that initially just sort of piqued my interest, through studying. I found more information than the professor like talked about, which is, like I said, is very common. So it's not. I love that class, and I'm not digging Professor Priya or anything. I love her, but yeah, I think that this specifically is something that is not popular, even when it comes to fungi in popular culture and how people are understanding it more. No one understands it. Like, yeah, I think it has spores or something. Yeah, but people don't even know that.
Yeah.
Some people think that mushrooms
So many
produce
ways
seeds.
that
you know?
spores can be spread
Yeah,
to,
exactly.
and that in itself is like kind of its own. Oh my gosh.
Yeah.
Do you have a fascination by spore dispersal as well?
I thought so. I actually didn't know about Mueller's drop until I made the presentation through my
Oh.
research. Yeah, and that blew my mind.
It's
that
interesting.
yeah.
Podcasts on spore dispersal and that in itself, like stinkhorns, for example, their spores are spread by insects. And then there's raindrops, like with the little bird's nests and puff balls. And that's just like a few off the top of my head, but it seems like that in itself is something that's not studied as well.
One thing that really interested me with like puffballs specifically is how they usually grow at the lines of the crowns of trees, because that's where the most rain will drop once it's raining.
And large drops
Exactly,
too.
yeah.
Accumulation.
Exactly. And it's those specific drops that they use to disperse their spores. Whenever the drop hits it, it like puffs out.
Mhm.
And that's true for literally every single mushroom. They all have their own specific environmental niche that they are filling. And those niches are not only not being studied, they're barely being found out that they exist in the first place. So I'm a huge believer that we need as many minds as we can when it comes to fungi in general. And if I can spark interest in any way, I did my job.
So if someone wanted to learn more about sexual reproduction, do you have any recommendations of other books or websites or places that you turn to?
If you're really interested in it, then I would recommend scientific journals. That's like the best way, honestly, because as I was saying, this isn't talked about very much. So this honestly may be the most condensed presentation on like YouTube currently of fungal reproduction. Honestly, there's a few that are that I got some information from, but most of it is very specific towards like specific species that you're looking at, or like general rules or something. There's not really something that encompasses like this is reproduction 101. And I was thinking about for example the monodikaryotic reproduction or the Buehler's phenomenon. Those are definitely something that's more advanced that I think would be interesting to talk about. But as specifically with that one, I've only been able to find scientific journals that talk about it. There was one documentary on YouTube about Buehler or Butler, whatever his name is. He was a very kooky dude. He apparently he was a professor at Melbourne, and he apparently got fired because he started giving his students psilocybin. Yeah. So
Was this in Australia?
or no, not Australia. It was Canada. Where is it?
Somewhere in
Yeah,
Canada.
yeah.
Because they have legalized it in some places, but I think it's just medicinal.
Yeah, nothing. Or it's in Canada. It is recreational now.
Yeah, it is
Yeah, you can go to some stores and do it. Yeah, pretty crazy. We're way far behind them. I think I'm everything.
So this illustration you did, is it the Devil's Urn?
I didn't really specifically get. I'm not sure. I just got like a sack. I just looked up sack fungi on Google. I just copied the first one that I saw. It was red.
Yeah. Oh, okay.
So
Yeah.
if that
Probably
helps.
up.
Okay, maybe. Yeah.
I've seen those here.
Yeah.
Central Texas.
I really need to study more of my Ascomycota. I definitely know Basidio more just because we use it more often. But there's a lot of pretty cool stuff. Yeah, this is just like a general cup of fungi, And even this is kind of wrong because they have two structures down here that are like a hub for the nutrients and stuff. But this is just for demonstrations of nuclei being fused. So yeah, as I was going to say earlier, you can see the mycelium that's going into the structure is still separated. Some of them are conjoined, most of them are separated. And then
it's just showing the different genetic strands.
yes, so this will be haploid. Yes, it is. Exactly. And this is where the diploid structure happens, where there's two nuclei per cell. And then in this cup fungi, the ascus is in the inside of the cup. And I did it purple, so you can see that the nuclei have fused at that point.
Oh, that's great.
Just a good example of like, you know,
It's not like a stadium party.
little worms that are dancing. Yeah, I did. Thank you. Thank you.
Yeah, how meditative is that?
It was nice, honestly. I had put my headphones on. Yep.
So if you have like, so you say you have mycelium, part of it has 7 cell. You have AB over here, AC over here. Sure. Like, do they compete within the mycelium? Then people want to do something.
Oh, no. The mushrooms are very free loving. You know. See, that's where it gets like kind of tricky.
It's like it would be an advantageous mutation for like one of the things to try to prevent cells different set of chromosomes.
Well, so but their goal is to reproduce at the end. So they don't really care who they're doing it with. And if one that is mixed already is seeing nutrient deficiencies or dry climate or whatever, they will die. But the mycelium that is connected to it that is still receiving nutrients and water and everything, they'll still be alive.
So like see if like eukaryotic cells that are like different in 70, 80, like they would they're kind of in conflict. There's some competition.
What do you mean conflict?
Well, like there's some limited amount of resources, and one of them gets to kind of C wants to reproduce itself, and E wants to reproduce itself. So like, I guess I would say that it's a competition between them for like have C try to suppress B or boost toxin or not toxic things.
As far as I know, I don't think so. I think that they're just trying to find whoever they're compatible with to get the next generation. You know, they don't really.
It's like after they've already.
Well, okay. So actually,
like they've already made it.
There has been some research. This is again something that I heard about the information. There has been some research to show that there is a preference for specific compatibilities over other. So when they are looking, it's not necessarily evenly distributed. It's kind of like a bell curve of compatibility. And the ones that are more compatible, they sometimes will outcompete the ones that are less compatible. But it's not necessarily that they're trying to kill it off or anything. It's just that they are more likely to reproduce. Not sorry. In a way, yeah, it's more of a passive thing than actively like producing enzymes or whatever to kill off specific genetic traits, unless it's like cancer. Yeah, sure. I think I missed some also.
3.72 inches of rain over 12 square inches.
So
let's talk about sexual reproduction.
we're about to see a whole bunch in the next couple days. Getting steamy. So I think there actually are some areas in Central Texas that emulate the Muscaria. Question mark. You know, they're asking if microclimates can be made in Central Texas for Amanita muscaria to grow. Amanita muscaria is mycorrhizal, so I think it depends on the trees that are growing. Yeah.
Mycorrhizal fungi are really hard to.
Yeah, there's been a few people like the Dutch Morel project that they're able to. Okay. Interesting.
Yeah. And there's
Saprophytes.
something maybe like a relationship with like grasses. Like, those grasses.
Yeah, I see that, because they're always on grassy
Yeah,
presentations.
yeah.
or whatever.
I've
Never seen
never
them.
seen this in like trees. anything.
Yeah, so some fungi, their fruiting conditions are the trees that they are growing with, and it's called mycorrhizae, and Amanita muscaria is one of those. So you would need the tree to grow the fungus, and maybe if you transferred the tree over here and grew it, and then put some spawn near there in a few years, maybe you could see that. But in the wild, I don't think so. I don't think it's very likely that we'll see that. Maybe in East Texas, maybe.
There's like a section. It's in the same category, but it's like not started. But I have seen them. They're really, really red.
Yeah. There's also the Razulas that I showed a picture of one earlier. Those are really common in Texas, and those are also very, very deep red. But they're missing the
Universal red.
yes, the universal veil and the warts that are on top. Oh, what is that word?
Yeah, I love that one. And
Yeah, the vulva. How do some hyphae become a stem mushroom and others a body? So that's going to be dependent on fruiting conditions. Depending on the gills or the other different parts. Ooh, so that's an even more advanced question. So that's going to depend. Okay, so think of it like whenever you are like a fetus is developing. It's going to be a very similar structure to the way that our fingers are able to be separated. So that is apoptosis that's going on there because it's a regular controlled cell death because otherwise it would just grow in a big clump. And the exact mechanisms, I couldn't tell you. It's probably some enzymes that are going on that are like killing cells as they're growing. But yeah, I would say it's dependent on the DNA of the mushroom. It's in the genetic information to tell this part of the mushroom to grow this way and this part of the mushroom to grow this way. In a similar way that we have DNA that says this is a hand, this is an ear, this is a leg, or whatever. It's all just in the DNA, and the DNA is each cell of DNA has like 6 feet of nuclei unwound, or maybe I think that maybe each chromosome has 6 feet. And each cell has like something like a lot. There's a lot of DNA to be read, and a lot of it is redundant and says to do the same thing multiple times for reasons like that. So just depends on the DNA.
someone on the YouTube is asking if you ever has anybody ever made spore art?
Yeah, kind
Like
of.
spore prints.
Yeah, just spore printing themselves are pretty hard.
Yeah,
Cool,
it's really
yeah.
artful. I like the idea that there's one cool project I learned about. Probably closer so people online can hear. There was a group. This was maybe like 6 or 7 years ago, like that did different kind of myco art. But they collected spores from all over. People could just collect them and send them in, and then they took them and put them in a weather balloon to kind of seed the clouds, which I thought was really fun. But also kind of like, I don't know what's gonna happen there, but I mean, I'm sure there's all those mix of spores already in the clouds. Although like mushrooms like the Texas star, which are only found in Texas, Japan, and now we know Taiwan, how like, if the spores don't find their kind of favorable environmental conditions or their mating types or their, if it's a mycorrhizal fungi, the right type of host, then they won't do so well. But yeah.
It's totally dependent. Yeah, it's super cool. Texas Earth stars everywhere. Or not everywhere, but how did it get there?
It's very abundant in this area.
Yeah.
But yeah, do we want to watch some spore dispersal videos?
One more question.
Oh.
Two haploid hyphae in the same clone join with plasmogamy. No, because one of the requirements for plasmogamy is for there to be genetic variation. The genetic variation can be very small. Like for example, you can have 2 spores that came from the same parent, and those can reproduce through plasmogamy. But if it's going to be a genetic clone that you cut out of agar or whatever, they will not fuse. I'm not sure if your question is referring to selfing, to kind of try and get specific traits out of an organism. But if so, the best way for that to happen is to keep the parents of the hyphae that you're trying to get, and just keep breeding it with the spores that it reproduces, because those will be genetically different enough that they can reproduce, but similar enough that you get the traits that you're looking for. If that's what you're asking for.
We're having the same ice the whole time.
Reproducing ice.
Not much ice. I have iron deficiency. Maybe that's why it's making more ice spores. Like, give this girl some iron.
Are there any more questions or anything? Anyone in the Zoom chat or YouTube, Instagram? Here? Anyone? I'm not seeing any on Instagram.